All right, welcome back. Here we are with our first speaker in this session on stage two. This is Bruno, who's going to talk about overcoming the challenges of animal-free growth factors. So, Bruno, over to you.
Right, thank you very much for your time today. First, I'll give a brief introduction about the company, what we do at Qkine, what we produce, and then I'm going to present the challenges faced whenever we produce animal-free growth factors and how we approach this. Then I will cover some of the vector design that you do at Qkine and how you use this to increase the expression in coli, and then some strategies to optimize the growth factors that we have been practicing. It's a company that's based at Murdoch House in Cambridge, UK. It was founded in 2016 as a spin-out from Michael Havoni's lab at the University of Cambridge. He works in the Department of Biochemistry, so he has a lot of experience with protein purification, structural understanding growth factors, especially with all his work that he has done with Activin A. Then several people started looking for him to acquire this kind of start the business. So we manufacture high purity animal-free growth factors, cytokines, and other complex proteins for life science applications and organic culture. So what are growth factors and cytokines? Growth factors, they are small signaling proteins that are survival, proliferation, and differentiation. And cytokines, they are small signaling proteins that coordinate the immune system, inflammation, or infection. And then why animal-free? Animal-free brings, for example, sterility. We don't need to worry about carrying over some pathogens from the cells. Scalability. Working with E. coli, it's much cheaper to work with E. coli, and it's much more scalable than working for like mammalian cells or insect cells. The ethical considerations, because we don't need to worry about how the animals are being raised and treated, and how we're collecting those components from the animals as well. And then, especially we have a lot, there's the benefit of having like the protein purity in the identity, because it has less chances of and carrying over some growth factors that we are not aware of. So overall, we at Qkine, we work use the expression plasmid inside the E. coli, we cultivate this, and then we induce those cells to produce our growth factors. If the purification, if it isn't, we isolate the inclusion bodies, we wash them, solubilize them, and then we set up refolding, purifies, and we have the bioactive protein. So first I want to cover a case study. This was done by Toby Howley, and it's basically they identify some TGF-beta contamination within the purified gene. So don't need to worry about it. I won't read the entire abstract, but we can see here that the purity varies between systems such as CHO cells or HEC cells. So there's a lot of variability between different batches. So they were worried about this. Strange results regarding DDF15. So they decided to compare the different batches to see if it was due to different. They realized that it wasn't TGF15. It was mostly done by TGF-beta that was like terminating the TGF-15 batch that they were using. And then they rise up that we need to have awareness about community, because we could be spreading false results and no one wants this. So TGF-15 is a member of it's a therapeutic target due to its role in nausea and vomiting during pregnancy. Unlike other TGF-beta growth factors, smart activation. And then when studying TGF-15 biology, the smart signaling was detected. So you can see here when they were using the TGF-15 from Pepotech, which came from mammalian cells, they started to see some of these activities which they didn't observe using GGF15 from Abican that was animal-free. So this just shows those results wasn't making sense because it was from the contamination per se, and then you can extend this could have those false results, and there was a lot of resource and time that is lost because of things like this. And using animal-free growth factors, it's better in this way because you won't have other contaminating products. But then the main reason why not everyone produces in this way is cells have all the machinery needed to properly fold the eukaryotic proteins. So some of them may require phosphorylation. And E. coli is not the best system to work with all of those, at least the standard E. coli. And then in addition to this, you also have that most of the growth factors, they have several disulfide bonds. And then just recap, the disulfide bonds happens whenever you have two cysteines close by, and then you have, if you have oxidizing environment, those sulfurs, they're going to bind by a disulfide bond. So the E. coli can do this because the cytoplasm is reduced, so it ends up that we don't have this disulfide bond, and then most of the times the protein will be insoluble and to kind of recover the properly folding of the protein. So this requires a lot of expertise, and fortunately we good. So before covering other topics, I want to give a special attention to the vector design, and sometimes it's just neglected, and it can bring lots of improvements as well. So for example, the codons, just to recap, is a 3 nucleotide sequence that corresponds to one amino acid. So you have that all the amino acids, they are coded by several different codons. And each organism, they have a certain preference for each of those codons. So we call this the codon usage. And then you can compare here that in blue, you have the codon usage for E. coli, and in red, the codon usage for human cells. So I want to give special attention to this part here, because you can see that there's a lot of difference between the blue and the red. So if you focus in those amino acids here, or in those codons here for AGA, AGG, which codifies for arginine, you have this preference for human cells. They prefer those codons. And then those ones are the ones that are preferential for E. coli prefers. So you can see the difference between those two. So for example, if you choose a codon that's rare in E. coli, it simply staggers all the expression, because then you're going to need to find that tRNA, and then you're going to bring the it will cause delays, and then this can decrease the expression. So for example, if you take the target A, you use two different strategies to optimize the codon. You see a lot of difference between those strategies. So for example, for strategy 2, we have this band here, which is the target protein. You change the strategy, you have the codon usage 1, and then you can see that it improves like 3 to 4 times the expression, at least in this case. In addition to this, we can also look at the vector backbone. So we can see here the regulatory gene, the ribosome bind site, and the coding sequence of the target that you want to work with. So if I have the RNA polymerase going to bind to the promoter, and then after it binds to the promoter, it starts transcribing the mRNA, and then when it reads the RPS, can bind to the RPS, and then this creates kind of a temporary complex between the ribosome and the RNA polymerase. The ribosome will end up protecting the RNA, so it prevents the RNA to degrade, and it also pushes forward the RNA polymerase. Helps to start the transcription to keep going, the transcription going. So this is the ideal case. But sometimes, if you have the promoter and the ribosome bind site too far apart, you won't have this additional protection from the ribosome. And then there are mRNA forming secondary structures that may stagger the RNA polymerase. So because of this, the RNA polymerase can't, and then it just gets blocked. So this ends up affecting your expression. So to avoid this, sometimes mini-cistrons, they are small codifying proteins. So they just kind of like, we know already that they are going to express well. So they force the early transcription translation coupling. What I mean is like they force the early formation of the polymerase being kind of coupling together to push forwards the expression. They also clear any secondary structures because then we already know that they're like properly, they express quite well. And because they are short sequences and we know how the RNA polymerase won't get staggered in there. So they clear those secondary structures. And then you improve the ribosome targets that you want to express. So for example, here you have for protein B, and then you can compare with and without the mini-cistron you have protein. And then when you use the mini-cistron, you can see that there is a huge boost in the expression. So it's much more advantageous to work with some if the material has like 1/10 of this. And then you have another strategy that I particularly like. So this one here, you have the shine Dalgarno, which will be recognized by the RNA polymerase. So it's a little bit contradictory to since you're adding those extra sequences, it pushes it. You increase the distance between the RBS, but at the same time you do this in a rational way because you have the shine Dalgarno bind to the anti-shine Dalgarno in the ribosome, and then the enhancer sequence. The idea is that it will bind to the S1 unit. Improves the complex between RNA and the ribosome, and you will regulate this distance by adding a poly A. So the poly A, basically, because it's all adenosine, they won't have any secondary structure in there, and you can kind of push the expression as well. The whole ribosome is completed, and then they start to synthesize your protein. So here in this with the expression of a protein C, we didn't have any expression with or without any mini-cistron. Even with tags, we couldn't see. So then, by adding the enhancer in poly A, you can see a massive boost in the expression of protein C. So it makes total difference in this case. And then now I want to talk a little bit about how you optimize the growth factors at Qkine. So for example, to analyze the structure, analyze the sequence of the target. So for example, first we try to identify all the amino acids that may be causing some issues. So for example, if you have hydrophobic in bulk amino acid that's exposed to the solvent, but it may cause some static hindrance in the folding. It may affect how it's going to interact. We may just mutate those amino acids and try to optimize the sequence. And then we also compare sequences from other organisms as well. So you compare the sequence of human, compare sheep, dog, pork. You can compare across all the animals that we have the sequence already. And then after you identify those conservation sites, you can see which ones they are more preferred, which ones they are not. Amino acids are not conserved, but they have some other options that you can use. This sometimes improve the thermostability. So then you analyze the thermostability and you compare with the wild type. The main point of analyzing the thermostability
information
about the folding as well. So if the protein is properly folded, you expect it to be quite thermostable. If the protein isn't folded very well, it tends to aggregate a lot early because you're going to have all the bulk hydrophobic coli exposed. So some examples of optimized growth factors that you have in-house is IL-34, activin A, TGF-beta1, and I will give particular focus to FGF2G3, which was done in partnership with Fernandes. So here we have
kind of like initial
screening about along some of the variants of FGF2. So they called FGF2
that combine the both
mutations. So here, for example, you have the arginine being mutated to tryptophan. That was
optimized to leucine.
So you can see how this affected the TM, that is the melting temperature. So from 1.3, it increased to 3.7. So not every single amino acid, but at least the ones that could be critical for the folding or for the thermostability. So this is the design approach that they use. And then you can see here that some of those were detrimental to the folding. So we have a decrease in the TM.
Pick the one that has a
positive impact. And then from those, they call the FGFT2. And then, I mean, you bring a lot of improvement in relation to the white type. So, for example, the white type
is the half-life of the
white type is 2 hours at 50 degrees. And then when they did those mutations here, they optimized the
65
degrees being the half-life of 2.6 hours, so it's much more stable. And then for FGF2G3, which is another round of those optimizations, then you can go up to 1.6 hours at 70 degrees. And then you can see how the folding was conserved along the time. Because you have the circular dichroism, you can see that the FGF2G3 is much more, the folding is much more conserved than the other one along the time. So you also compare the activity of those. So in here you have the FGF2 wild type, which is in green, and then you have the optimized version, the G3, which is in black. This is day one, so they have similar activities. It's 3 picomolar and 5.7 picomolar. But then after incubation for 7 days, in liquid media, they found out that the activity for the white type simply dropped a lot.
Don't have any more activity.
But the thermostable version is completely fine. And then it remains bioactive even after those 7 days. So overall, here we talked a little bit about the benefits from utilizing animal-free growth factors. I want to highlight the importance of the upstream because sometimes we simply neglect all the optimizations that can be done at the DNA level before going to the bacteria. And also, it's just a small example of some of the things that you have been trying to optimize at Qkine to try to raise the standards. So we don't want to just keep delivering the growth factors. We want to make them more thermostable, more active in a way that you bring the innovation, but you're also trying to keep sustainable without any animal-free derived components. So thank you very much for the time and the attention of everyone. If you have any questions, you can just ask. If you can't cover this now, you can just send me an email and then we can talk.
All right, brilliant. Thank you so much. I'm going to dive straight in with a question of my own, if I may. I'm interested. I mean, it's interesting that you mentioned there at the end about this idea of upstream and downstream, and the sort of purposes. But can you kind of dig into that a bit more? What's the impact here? Where do you see this delivering the most benefit for the kind of sector as a whole?
That's really great point. Because, for example, let's say that we saw several talks. So we are scaling up the expression of a target. We are taking from like 1 liter scale to 100 liters scale. We can optimize the expression, but at the same time, if let's say the code optimization that I showed before, that's not the most efficient is being used, instead of having like 100 liters, we could be working with 20 liters and it would have the same yield, but you would have less costs. You wouldn't produce that. So I think it's quite important to bring attention to this, because at the same time, you can also change the vectors. You can try to make that are not expressing well, so you can just boost with this some of the tweaks in the DNA.
Okay. Yeah, no, I mean, I think that's really important. Just to dig into a kind of practical example here, because obviously there's a lot to think about in what you're doing and a lot of different choices that you have to make along the way, I suppose. How do you, for example, decide on fusion partners or tags for, solubility and folding, for example?
Mm-hmm. I mean, tags are useful because, for example, when you're using the N terminals, you know that most of them, but at the same time, you end up having some scars. So whenever we separate the tag from the protein, it may leave some extra. If you don't remove the tags, you don't quite know if they are going to affect the activity. So sometimes you may be working with a protein, then you have tag. And just as soon as you remove the tag, the protein just collapses. So that's why we avoid work with any kind of.
Yeah, I mean that sort of leads me on to my next question. Like, when you're going through this process, what were the kind of hurdles, I mean, kind of structural or biochemical hurdles that you faced along the way, and how did you deal with those?
Mm-hmm. I mean, to be honest, once you have the protein is expressing well, if the protein is soluble, you remove a lot of the headaches along the way. The main soluble, because then we need to find a way to properly fold the protein, without using anything from the cell. All the different bonds, you need to make sure that the protein is assumed it's properly folding. So for this, we have to optimizing the redox conditions, which buffer you're going to use, which salts.
Yeah. No, absolutely. So tell me, I mean, finally, if I may ask, what's next? Where do you go next with this? What's the plan?
So
with all this work you've been doing, what's the next steps? What's the next bit of research? What happens after this?
I mean, there are several growth factors that are not produced in E. coli because of the challenge that they face. For example, some of them have several disulfide bonds that we don't have. The E. coli is not the best machine to make those. So I would say that the next challenge is getting all those growth factors that we don't have now animal-free supply and make them available to other researchers. We also want to tackle all the kind of more difficult problems with those, because some of those growth factors as well, they are not thermostable. They aggregate quite easily. So we always try to push this and make it more efficient.
Okay, sounds good. I mean, we'll be looking forward to seeing more on that. Thank you, Bruno, so much for that presentation, for joining us here today. If you have more questions for Bruno, do find him on the platform and doubtless elsewhere as well. And you can, take up any more detailed questions with him there. But Bruno, thank you very much. We're going to go off now, and I will bring our next speaker on shortly.
Thank you.